And so food systems transformation it starts with biodiversity. There is no other way for us to be able to create the crops of the future without the diversity. So being able to tap into the diversity in ways that allows us to be much more precise and faster than we used to do is something that we are going to be discussing in this meeting. And I think it's an important call. We need urgency, we need action in really tapping the potential of the diversity that are today in the gene banks or Exito.
The global system for plant genetic resources for food and agriculture, which includes mechanisms such as the International Treaty, the Commission on Genetic Resources for Food and Agriculture, and the Global Crop Diversity Trust, is very important today. This is because our food systems are increasingly under threat.
Gene banks have evolved over the past 100 years, and they're resources that are clearly underutilized. They can be utilized more effectively in the 21st century if we could build networks of investigators with appropriate support to go in and ask important biological questions that deal with quality of the genetic resources, climate change, disease pressures, soil changes. Those are all going to be really important, and those will help identify the tips of the iceberg that will be valuable for crop improvement.
Well, I see two areas mainly. One is high throughput genome sequencing, which is invented for having medical genomics in place for diagnostics in human, but we can take advantage of this to really unlock the entire crop diversity that is hosted in gene banks through throughput sequencing, because this will give us access to all allelic diversity that was collected in the past and that is accessible and available today for the breeding and research area. The second area of technological innovation at the moment is in the area of the new breeding technologies in gene editing, because this provides, in theory at least, a possibility to bring allelic diversity into crop material more quickly. So alleles that we discover in genetic resources that are not directly amenable to the breeding cycle can still be a very rich source of allelic diversity in context of climate change.
We can start now breeding on DNA only without phenotypes, and that helps us to reduce the time that we need for one cycle of selection. So we can now have, like, for example, in corn, we can have like 3 cycles of selection in 1 year instead of only 1. And that, of course, helps us to accelerate free breeding and bring the genetic resources up to the level of elite germplasm, so we can use them also for crossing. That is certainly one of the best like achievements that we can get on quantitative traits.
To find those gold nuggets, it will take more advanced technology to go in and do the trait analysis that we call phenotyping. To go in, and whether it's phenotyping associated with a plant characteristic like height or fruiting or other characteristics, tillering, or also characteristics that would be biochemical, nutritional traits, or anti-nutritional traits. Having improved phenotyping technologies in the 21st century to go and complement with the genotyping technologies and the data management will be critical to find those nuggets.
We have this quite wide range of emerging technologies that are accelerating in pace from AI to genomics to remote sensing, robotics, phenotyping, and so forth. But it really are big challenges that to harness these and in a manner that has an impact on the gene banks, has an impact on breeding, has an impact on delivery, but also we have a really major challenge about how do we democratize access to those technologies that impacts on the lives of those that do not have access to even most basic technologies. So there's a technology access gap that is inherent in the challenge that we face. Even though we have all these wonderful technologies, we really need to harness them in a manner that is equitable.
it's pretty clear that digital sequence information can contribute to breeding processes, can contribute to the conservation of plant genetic resources, to the characterization, can contribute to the development of new products. On the other hand, there may also be risk involved. These are issues like the safety of new plant varieties, which obviously has to be ensured. And there is, of course, also the risk that access to digital sequence information is being limited, which could hinder research and development in the future.
To unlock the genetic diversity in gene banks is really not that expensive, even if you put it into the context of medical genomics. So it is the question of putting it into the right context. We are talking here about food security, and we talk about it in the global context. So I think the investment is not too high. The technology is ready and there. We could do it, but we need international cooperation. We need the willingness to cooperate as a human community.
As important as for us to deploy these technologies is how we do it. As CGIAR, we are the largest global public goods in agriculture. So we host, over 700,000 sessions in our gene banks across the globe. So we are really, in a very good position to help to make sure that this is public access, that we apply these technologies, we make sure that countries have access to it, even the countries that might not be able to today invest.
I think there's a range of policies that are likely to be necessary to consider in relation to technologies. One in particular that we will almost certainly have to grapple with is the question of data. We enter an era of very data-intensive agri-food systems, and questions of ownership and control of that data and the use of that data for improving people's lives. Our policies need to guide that through incentives and disincentives.
One objective is obviously to keep open access to digital sequence information because it is essential for research and development in the food and agriculture sector. It is essential for food security ultimately. But obviously, we also want to make sure that the benefits derived from digital sequence information are shared equitably and fairly, including with indigenous communities who have been conserving for many, many centuries plant genetic resources. So the decision taken by the Conference of the Parties on digital sequence information tries to achieve this balance, tries to keep open access for, in particular, the academic and the research sector to digital sequence information, and on the other hand, requires monetary benefit sharing from commercial users of digital sequence information, who in the future would have to provide part of their benefits, of their profits or revenue to a global benefit sharing fund, the so-called Kali fund.
People have to be co-developers of the solutions. So the gene banks in the developing South will not be content to just be recipients of handouts, to be just recipients of solutions. I think it behoves the international community to strengthen institutional and human capacities in these national gene banks.
Crop diversity is important in various respects. Number one, to cope with crop diseases. Number two, to deal with human health, because humanity needs diversity in the foods which are produced and shared and are accessible to people, be it through shops and markets and so on. And we need crop and agrodiversity for a diverse environment in which biodiversity is protected. So these three objectives are absolutely critical for planetary health.